I'm a principal scientist at PacBio. I'm going to discuss variant detection with PacBio HiFi Reads as it pertains to genome in a bottle benchmarks and the precisionFDA True Challenge v2. The typical goal in sequencing a human genome is to identify the differences between the individual being sequenced and the standard reference, and these differences are called variants. There are three main classes of variant: single nucleotide variants, where a single base is changed, such as a C to G, shown here. Short indels, which are insertions and deletions of 1 to 49 base pairs. For example, this deletion here, where the individual HG002 is missing a base present in the reference, or this insertion where the individual has bases that are not present in the reference. And finally, structural variants, which are indels greater than or equal to 50 base pairs, or inversions and translocations. PacBio HiFi reads are long and accurate DNA sequencing reads. Typical read lengths are around 15 to 20,000 bases, which is about 100 times longer than a typical short read length. And the accuracy is over 99%. These HiFi reads provide the most comprehensive view of human genetic variation. Compared to the popular short read sequencing approaches, HiFi reads detect more single nucleotide variants and indels in the difficult to map regions of the genome, and long insertions and deletions and structural variants genome-wide. A typical individual has, by count, more smaller variants, around 5 million single nucleotide variants and 3 million bases and indels, but by number of base pairs affected, most of the variation in an individual genome lies in structural variants. To show some examples of single nucleotide variants and indels in difficult regions that are better detected with HiFi reads, you can look at this gene STRC. This is a congenital deafness gene that has duplicated copies elsewhere in the genome. So there's the functional gene copy and then some other segmentally duplicated regions that originate from the same original sequence. And thus it's not possible with short reads to confidently map a read to either this functional gene copy or the duplicated copy elsewhere in the genome. Well, with HiFi reads, you're able to span from the unique sequence all the way through the duplicated region and identify variation in all exons of this gene. STRC is only one of many medically relevant genes that are better covered with HiFi reads. There's been published lists of medically relevant genes that are not well covered with short read sequencing, and among the 193 mentioned in this paper shown on the left, 152 of those are fully resolved with HiFi reads. Moving to long insertion and deletions and structural variants genome-wide, these large variants are better detected with long reads. This is an example of a 1,733 base pair deletion, where it's clearly delineated with the HiFi reads on the top, and on the bottom the short reads have ambiguity of mapping to this repeat that spans the deletion, and it's not possible to tell whether these reads originate from this copy of or this region of the genome or somewhere else, and thus the deletion is not able to be detected. Those are just anecdotal examples, but in terms of rigorously measuring the performance of variant calling, you can look at genome in a bottle benchmarks. The benchmarks are for 7 human genomes have been established by the Genome in a Bottle Consortium, which is a group of academic, corporate, and government scientists led by the National Institute of Standards and Technology in the United States. It's part of the United States Department of Commerce. And Genome in a Bottle selected 7 reference samples, sequenced them deeply with multiple technologies, and generated truth benchmarks. The benchmarks then can be used to evaluate call sets generated with another technology. For example, here's showing a cartoon of a call set, and going through those calls, you can then classify something as a true positive, meaning the call set matches the benchmark, a false negative where the benchmark has a call that's absent from or has a variant that's absent from the calls, a false positive, or a false positive where the calls have something that's not in the benchmark. And from these true positive, false negative, and false positive measurements, you can calculate metrics called precision, which is a measure of the false positive rate, and recall, which is a measure of the false negative rate. In 2007, Genome in a Bottle released the version 3.3.2 of its small variant benchmarks that were based on multiple short read sequencing datasets. This benchmark covered 85% of the human reference genome, but only 24% of the difficult regions of the genome that fall into the category of segmental duplications and low mappability regions. And this is because the short read technologies used to build the benchmark do not categorize these regions well. In late 2019, continuing into 2020, the consortium updated their benchmarks. Specifically, they started with updating the benchmark for the sample HG002, and increased the percentage of the genome covered from 85% to 92%. Much of this increase was in the difficult regions of the genome, thanks to the inclusion of HiFi Reads and linked read datasets in defining the new benchmarks. The percentage of the segmental duplications in the low mapability regions covered improved from 24% to 54%. Genome model then moved to produce improved benchmarks of its other samples, starting with HG3 and HG4. But rather than releasing these benchmarks immediately, they launched a contest called the precisionFDA Truth Challenge. Groups are provided with reads for HG3 and 4 from the Illumina NovaSeq, the PacBio Sequel II system, and the Oxford Nanopore PromethION, and they were invited to submit call sets that were then compared to the blinded truth sets and ranked for performance. The results of this contest were announced in August of 2020. In total, there were 64 entries. 24 of those used exclusively Illumina data. 20 of them used multiple technologies. 17 of them used exclusively the PacBio HiFi data, and 3 of them exclusively the Oxford Nanopore data. Ranking them by overall accuracy, 25 of the top 26 entries, including all of the top 12, used PacBio HiFi reads. The very top performing call sets were multi-technology sets, shown in gray here. The next sets, the highest performing single technology sets, were PacBio HiFi reads, followed by the Illumina reads, and finally the Oxford Nanopore reads with the lowest performance. The overall highest performing call set was a call set from the Google Deep Variant caller using PacBio HiFi reads. There are also high-performing deep variant call sets for the other technologies. You can see the Illumina Deep Variant set was the second highest scoring Illumina set. It far outperformed the de facto standard Illumina GATK call set, and that outperformed the Oxford Nanopore Deep Variant call set, which was the highest performing of the Oxford Nanopore sets. Looking at the performance in a different way, the PacBio HiFi Deep Variant call set had 13,067 total errors. Again, this was the fewest of any single technology call set. The Illumina Deep Variant call set had 2.5 times more errors, primarily due to false negatives and false positives single nucleotide variant calls. Illumina GATK had 5.8 times as many errors, and the Oxford Nanopore Deep Variant call set had 30 times as many errors, primarily due to indel performance. This improved ability of PacBio HiFi reads to detect variants has been used not only in benchmark genomes, but then taken to disease genomes and used to identify pathogenic variants that had been missed with short-read sequencing. This is showing 2 examples of that in Mendelian disease cases where there were exonic deletions that had been missed with short reads and were identified with PacBio reads. Because of this performance in these initial examples, PacBio HiFi reads have been adopted by large consortia studying hundreds of human genomes. In the disease and cancer research area, the SolveRD group has adopted PacBio HiFi sequencing for 500 individuals. And in population genetics, Hudson Alpha Discovery and the Human Pangenome Reference Consortium have adopted PacBio HiFi reads for thousands of individuals. So in summary, PacBio HiFi reads are highly accurate and long DNA sequencing reads. They provide the most comprehensive view of human genomes with high precision and recall for all classes of variation. The HiFi call sets achieved the highest accuracy in the precisionFDA Truth Challenge v2, for which more information is available at the blog linked here. And HiFi reads are being adopted by large efforts in the rare disease and population genetics space.